This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 598, recorded on April 3rd, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from a hospital out on Long Island, Daniel Griffin.
Hello, everyone.
Welcome back, Daniel.
Thank you. Happy to be back.
You're not in a parking lot, actually. You're in a stairwell, right?
Actually, I went out to the car. I was worried about, I think I told you in the stairwell, is that there's this intensive care unit, which has 20 beds, people intubated, and basically there's so much of the SARS-CoV-2 in the air that you're wearing an N95 respirator the entire time. So I had, snuck out into sort of this back stairwell, but when I was talking to Vincent last time, I noticed that there's a crack under the door. So I'm not sure how safe it was for me to be breathing there without my respirator. So I left the building for safety purposes.
Okay, great. Also joining us from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent and Daniel and everybody else.
And from Austin, Texas, Rich Condit.
Hi, everybody. 71 degrees, 22°C and raining.
And from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's sunny and supposed to get up to at least 60 today. It's 59 right now.
Okay, Daniel, give us our pandemic update.
All right. I guess first off, Dixon, it sounds like you survived. We have read a lot of people email me. They were worried. They were worried about Dixon, so he's better. This is good.
No, I'm hunkered down.
So let me give my my talk from the trenches, so to speak. My update. I know a lot of clinicians listen to this and they like it to be to the point. So I will. I'll hit some highlights. The first thing is actually, this is new. We'll talk about a little more in terms of prevention. And I think people are going to start seeing, at least in the New York area and more widespread, a recommendation that people wear face masks to try to improve the flattening of the curve that we're accomplishing to some degree with social distancing. And early on, a lot of us really were cautioning people not to wear masks, in large part because they're at limited supply. And so as people already know, we've experienced shortages. So part of our recommendations regarding masks early on were to help protect healthcare workers from the shortages that we're now experiencing. But you'll start to notice, and actually I was talking to interviewer on NPR last night about what are the role of masks and when does it make sense? And actually, I think now we're at a point where it makes sense to wear masks, to recommend them. And I'll talk a little bit about that. And I was enjoying This Week in Virology. Ian Lipkin was talking about some research on masks. And there is research on masks and a certain time in which they can be helpful. And it's interesting. They have to be worn correctly. They have to be used correctly. And interesting enough, they tend not to work particularly well until the people using them are aware that there's a pandemic or an issue going on. And so a couple of the studies that I think now will be getting attention are studies where they show that during a major disease outbreak, if people wear masks, they can actually not only protect themselves, but there can actually be a decrease in transmission to others. So you're doing something nice for yourself and others. And but I try to point out that when we make recommendations as physicians, as healthcare workers to the public, we like to point out that a mask can be worn properly and it can protect us, or it can be worn improperly and can actually increase your risk. So I like to explain that. And I think we've had an example of an improper mask usage on an airplane where who was that on the airplane where the woman next to them offered?
That was me.
So Rich, I remember your story, and the woman next to you, would you like a mask? And then there were people, you always see them do is they like to take their hand and touch the front of the mask. They like to lower it down, often put it on their chin, and then they feed themselves peanuts or whatever, or they slide it up and put the peanuts underneath. And so the way a mask can protect us, and I think people need to realize this, you put the mask on, and now the mask is doing two things. It's catching all the moisture from when you speak, cough, or sneeze. So the inside of it is now becoming filthy. So not something you that want to then slime down onto your chin and slime back up. And the other is that should someone get within that 6 feet zone and cough or sneeze at you, it's protecting you from the cough or sneeze. But if you put your hand on the front of the mask, now you've contaminated your hand, and all the sort of dirtiness the mask is supposed to help you with is now on your hand. So, a mask is like a pair of underwear. You put it on, and then you take it off and throw it in the wash or the trash, but you don't mess around with it, put it on your chin, etc. So we're now starting to recommend, in addition to social distancing, the proper use of masks. The next thing, as far as prevention, we've got a big trial. I was emailing with Vincent earlier. Our idea on the Plaquenil, the hydroxychloroquine, has been that this, like most other antiviral therapies, is most important in the first 48 hours of symptom onset. So we've got a couple trials, Patch 1, 2, and 3, and these are prevention and treatment COVID-19 hydroxychloroquine trials. And so the Patch 2 and 3 we're doing up here in the New York area. Patch 2 is going to be acute onset treatment. You've been sick for a day or two and you start on the treatment. We're hoping it decreases that hospitalization rate from 20% down to a lower number. And Patch 3 is going to be healthcare worker prophylaxis. It's going to be healthcare workers taking it to hopefully keep them from getting sick on the job. And unfortunately, as people have probably heard, a lot of healthcare workers have gotten sick here in New York and other parts of the country. So we're working on that a little bit. The next is, I know a lot of people are seeing patients in the outpatient setting, so we try to give some guidance there. And still sort of similar guidance, but maybe a slight update we have now is something that's we're going to be getting into. Maybe we'll discuss a little more as we go further, but the potential role of appropriately dosed and timed steroids. And this was an issue early on, right? No steroids. Steroids are bad. It'll kill everyone. And I think that came out of some ideas that particularly during the first week, when you need your immune system to get that viral load down, that shutting down the immune system looked like it was a bad idea. But there was a study actually that, it was like one study changes clinical practice in the time of COVID-19. It used to usually take a couple. And this was the title's interesting. I would like to rewrite the title, but the title was Early, Low Dose, and Short-Term Application of Corticosteroid Treatment in Patients with Severe COVID-19 Pneumonia, a single center experience from Wuhan, China. And what they did here is they started steroids when patients had respiratory rates of about 30. So what we describe is you're taking care of a patient in the outpatient setting. Their first week, they feel crummy. Actually, I enjoyed Ian Lipkins. He's like, I had something. It was just a URI for the first week. Then I got COVID-19. It's like, well, actually, COVID-19 acts this way. The first week is punky, so to speak. As people describe it, it only feels like you got run over by a truck. But the second week is when it comes back and drives over you repeatedly. And so sometime during that second week, when you start getting low oxygen content, when you start having trouble breathing, and here the people were hospitalized and breathing in the 30s, and they would start them on Solumentrol, so an IV intravenous steroid. And this would decrease the oxygen requirement. It was actually keeping some people from needing to be put on a ventilator. And we're actually seeing a little bit in the outpatient experience now is when people get to that point where they're starting to increase their respiratory rate. Sometimes 5 days of high-dose oral prednisone is keeping them from requiring hospitalization. So, everything we can do to keep people out of the hospitals is great. But sometimes those people end up in the hospital, and then we're seeing, this is another sort of change in treatment. We're seeing people reconsider the role of steroids. And so when they hit this point where they've gone from just a low oxygen requirement up to requiring more oxygen, we're actually a lot of centers starting steroids, just 5 days, a limited sort of timed and appropriately dosed. And we're seeing that people are able to avoid requiring intubation. We're able to see them get through this critical window, which is fantastic, like from a clinical standpoint. The other, I think we talked, this is something I'm pretty excited about, and I was on the phone with a bunch of clinicians over the last few days. But and I enjoyed Immune. I was listening to Immune. I love the theme music on Immune. It's calming. So I'm going to listen to Immune more often. So thank you for that podcast. But this cytokine storm that we're seeing, and I was discussing this with another person, and they were saying, "Oh, I just spoke to Robert Gallo, famed for the HIV discoveries, and he thinks IL-6 is really important too." So I was like to be on the same page with someone like that. But what we've started doing at a number of the hospitals in the area is we give the steroids first, and then if the person continues to get worse and it looks like they're heading towards requiring intubation, we'll do a cytokine storm dosed IL-6 receptor inhibitor, tocilizumab. We nickname it Toci. And we've actually seen some really impressive impact on the natural history of what we were seeing. I'd give a list of these people are going to be required to be intubated, and then they'd bounce back. They either wouldn't be intubated, or if they had just been put on a ventilator, instead of the 2 to 3 weeks, within a day or two, they're back out on the floor. And I'm discharging some of those people today, which is an amazing, you don't need a statistician when the impact is this dramatic. But this is, I think, really important. A lot of centers that are studying the IL-6 inhibition were not giving steroids. That was actually a reason why you couldn't be enrolled in the trial. And so at Columbia, where we can get a lot more labs, if you give tocilizumab, which is an IL-6 receptor inhibitor, it blocks the ability of IL-6 to interact with the receptor, and the IL-6 levels shoot up. And then 2-3 days later, the people suddenly crash too poorly, die in some cases. So in our experience, it looks like that giving the steroids prior to the tocilizumab is critical. So this will be interesting to see as we get real data. I would say the plural of anecdote is not data; it's anecdotes. So we're building an experience here of dozens of patients now seeing this impact on natural history. So it'd be nice to see this validated in some proper controlled trials. But this has been, for me, like a reason not to be as demoralized each day at work, seeing that we're actually doing something which seems to have a very obvious impact on the natural history. The other, which again, this is, keep things that initially people said, never do, it'll kill everyone. We're changing our tune and saying, do it at the right time in the right patient. And this is anticoagulation. We used to say, don't do that. These people are headed to DIC. But this was actually a decent study with hundreds of patients. And it was anticoagulant treatment is associated with decreased mortality in severe coronavirus disease. And so there's actually been now patients that have a particular blood level, D-dimer, that gets, let's say, 5 to 7 times above the normal limit. That these people on a low molecular weight heparin are actually doing better, because it looks like with this disseminated intravascular coagulopathy that occurs, that they actually get small clots everywhere, and that this is associated with better outcomes. So we're seeing that there. Those are the big ones. Those are the big, I'll say, clinical updates.
Can I interrupt just a minute? There's a very low death rate supposedly going on in Germany right now. Do they do any of the things that you've been talking about?
So I'm always hesitant when I hear these low death rates, because as I tell my wife, it takes time to die. And so when you initially see the numbers increase, which I think we're starting to see them ramp up in Germany, people don't die right away. And in the early experience, it was about 25 days from onset of symptoms to death. And then in some areas, we've seen that shorten to maybe 14 to 17. So you always have to watch the death rate as the number of case rate is accelerating, and see sort of where it pans out.
Daniel, how does it look compared to last week? Up, down, or the same in terms of total patient number?
We are still accelerating, and that is disheartening. We are accelerating to the point where I think the current forecast is that in about 7 days, we are out of ventilators. Basically, Yeah. So, we were all excited about the big ship that came into New York. I think so excited that everyone ran out and forgot about social distancing. So we're going to get a little surge there. I wanted to go see the ship too. And I think there's 15 people that are now being taken care of on this huge ship that can supposedly take care of 1,000 people. And there's a ship out in California with about 10 or 15 patients as well. So these huge ships have come in and they look beautiful. And I think Michael Dowling, the head of the whole Northwell system, was quoted as saying, yeah, it's kind of a joke. It's frustrating. Like, here's this resource, but I don't know what the bureaucracy or what is happening, but we're still drowning here. When we say we're still accelerating, every day there's more patients being admitted per day than the day before, more patients coming to our ERs than the day before. And that rate is still increasing. And at some point it will stop increasing. And then maybe in the next 2 to 3 weeks, it'll be a steady stream in the New York area, which is somehow more manageable than just this accelerating. But at the same time, we're going to be past the point where we have resources, and that's a huge challenge.
What do you do when you run out of ventilators?
So the first thing I think we've talked about before is trying to get 2, 3, 4 people on a ventilator, trying to somehow basically split the tubing. And then, and you could do it. People have done this. And I think I've said before, it's better to have a quarter of a ventilator than none at all. And it changes a little bit how we manage the ventilators, because normally we would set for a person on a ventilator how many breaths per minute, the amount of volume that we're getting, and the oxygen level we want. This way, you've sort of got to come up with something that works for 4 people. So you might use what we call a pressure support, where you're sending a certain amount of pressure out to everyone, and probably using a higher level of oxygen than you need, but sort of the highest you need for the lowest person in the group. So there are ways to do this, but yeah, we're getting into a much less than ideal territory. And I should jump in, actually, at this point, we've already run into the issue that the lab capacity. Early on, everyone was excited to do so many blood tests. And last night I was on the phone with one of the hospitalists out here, basically saying we've really got to limit the number of lab tests we order, because we just can't get them back in time. Lab workers are sick. Lab workers are over capacity. And if you order everything you want, you're not going to get anything. So we've tried to say, what tests do we really need? And let's all agree to work together. We can't have one doctor ordering 15 tests on every patient every day, and then the rest of us wait until 4 p.m. to get our morning complete blood counts.
Yeah. Last week, after we were doing listener email, we had the question, what are the discharge criteria for COVID-19 patients? And then kind of related, because I'm asking for a friend, really. If a person has been at home and is sure they had COVID-19 but was never tested, when could they conceivably go out without fear of transmitting?
Oh, no, so Kathy, great questions. I feel like, these were seated there. They put the person in the audience to ask that question. So thank you, because these are huge questions, and it is changing the paradigm. For discharge, we are sending patients out that still have fevers, that still are requiring oxygen, but are getting better. And so the criteria really is that they've made it either through this critical second week without having a bad outcome, or they've started to improve enough during that second week. And we can predict that. I was sharing some data that will try to get published when somebody stops, and has time, but where we use this neutrophil lymphocyte ratio, this NLR. And once we see that ratio going down, and some of the stuff I showed Vincent, we're trying to get published, but the people try to publish it are now being pulled to the trenches, and then another person got sick. And so at some point it'll see the light of day. But this neutrophil lymphocyte ratio, if you look at people that have levels of, we'll say, less than 6, people in sort of the 3 to 6 range, which is higher than 6, really very discriminatory, and p-values of 0.0074, which means that it's sort of obvious to see in daily practice. And if their NLR is on the way down, if their oxygen requirement has been dropping down and is now 4 liters of oxygen or less, even if they still have fevers, but they're feeling better, we try to get them out, before they get something else, before they get sick. And also so that we have room for all the people coming into the emergency rooms every day. So that's the discharge. But we tell them when they get discharged, you're probably still shedding virus. And there's such limited access to testing now that it's not going to be easy for us to document that you're negative. So the CDC guidance has gotten a little bit, I'm going to say, more relaxed or softer. And so they've basically said as long as it's more than 7 days since symptom onset and you've gone more than 72 hours without signs or symptoms, so you got to be off that oxygen. You shouldn't be coughing or sneezing or running a fever anymore. You should basically feel better. Then the level of virus shedding is probably low enough that you can go back out into the world. But then we've got the science, right? And the science tells us that, you're sending people out in the world that are still shedding virus there. And I understand the recommendation because we're in the middle of the pandemic. And so these are not the same criteria that we were using early on where we said, and that China was using in certain areas where they said, you've got to get 14 days and then another 3 days of no symptoms. And then you can get 2 tests. And if they're negative, you're good to go. So what I tell people is I actually give them the full story. I say, here's the CDC recommendation, but just to let you know, you're probably still going to be shedding some virus. The only way to know is to wait longer. And so, a lot of people are on home quarantine anyway. So if it's a retired person who's, hanging out at home, I say, you know what, if you can wait 14 days, please do. Because the science suggests that duration of shedding varies between people and varies between severity of disease that you might have experienced.
Okay, so if a person is still coughing, then you would say they should wait 14 days from when they stop coughing.
So when the symptoms stop, you want to wait at least 72 hours without symptoms before you consider saying you're good to go.
Daniel, what's the meaning of this executive order from the New York State governor about Plaquenil?
Yeah, so I'm glad you bring that up. Just yesterday, I think it was, his son Andrew. I'm old enough to remember Mario, quite the statesman. But so his son Andrew, who actually is, I think, really risen in this and done a great job. He came out with an executive order this week saying that there shall be no pharmacists filling prescriptions for Plaquenil, hydroxychloroquine, for COVID-19 indication in the outpatient setting. And it really, I've sort of been back and forth and talked to people about the studies out of Marseille and the one study out of China, in many ways in our mind being a string of anecdotes that with sort of carefully selected patients to support the role of these drugs. And what ended up happening is the press grabbed on a lot of this hydroxychloroquine, Plaquenil data. And there was really a run on, and I like to say, like, everyone and their grandmother needed it. And I say that because what would happen, and you get a call from, Joe Smith, and he feels horrible, and he's got COVID-19. And he wants a prescription, but then what about my wife? What about my children? What about my, grandmother who's living here with us? Doesn't everyone need it just to be safe? And so very quickly, the tablets, the Plaquenil dosages were so depleted that patients that were on it for FDA indicated reasons, such as lupus, autoimmune diseases, they did not have access to their medications. And so without really, compelling data that this made a big difference, I think that's what prompted this executive order saying, and we'll know hopefully in a few weeks with these big trials, let's know, when it works and who should get it. And then actually our trial patch 2 and 3, we're working with the FDA to get an indication. And our thinking is that the indication will be like early onset within, 48 hours of disease onset, outpatient treatment is going to make a difference, and also prophylaxis. So
anything else from the crew before we let Daniel go?
We had one question about whether the use of CPAP before ventilators could help reduce the need for ventilators?
Yeah, the stuff that came out of China early on, they basically said, non-invasive mask ventilation, so NIMV, and that would include CPAP, so continuous positive airway pressure, or BiPAP, which is another modality. Just to explain those for everyone listening, the CPAP is when you take a breath, you're sucking air in. What the CPAP does is it increases the pressure, on the outside. So when you take a breath, it actually gives you an extra amount of pressure, 5, 10 centimeters of water amount of pressure. Sort of makes it a little easier for you to work. You can hook up oxygen to some of these, and it can be a bridge, and was a bridge to a ventilator, or often kept people off a ventilator before. The other is a BiPAP, and this is a combination. When you breathe in, there's a certain amount of pressure, but there's also a certain amount of pressure when you breathe out, which helps the lungs stay open. And, prior to COVID-19, these were techniques we used quite widely to potentially keep someone from ending up on a ventilator or delay the time when they might need to go on that. The couple things that we're seeing in the China experience, and we'll have to see if it's any different here, but there's such a rapidity for when people decompensate and they go from requiring 100% oxygen to needing to be on a ventilator, that putting them on CPAP, BiPAP never really seemed to make much sense. And by the time they ended up on a ventilator, the experience, and of course this is like not a controlled trial, the experience, what was communicated was that they did worse, and you were better off just to intubate them early, for patient outcome, but also there was an aerosolization that's occurring. So if you use CPAP or BiPAP, you generate very small particles, less than 5 microns, which is the particle size at which you can actually float around on currents of air in the room for up to 30 minutes. And anyone who dares breathe in the proximity will breathe in the SARS-CoV-2 and get infected. So there are a couple recommendations why we didn't want to do that early on. Now we're at a point where often the entire setting is airborne contaminated. So there's the aerosol of these tiny, less than 5 micron particles floating around. So the risk to healthcare workers is less because we're all wearing N95s in these settings. But we're still not sure if it'll improve patient outcomes.
I wanted to just ask quickly about the rationale or mechanism of using steroids. I don't quite understand what that's supposed to do theoretically.
Yeah. I was thinking before I made this call that we should get Stephanie or one of the co-hosts from Immune to come on. But the idea would be this. The first week of the illness, it's all about the virus, right? The virus is replicating, the immune system is starting to ramp up. And then usually when we see them during this critical period of decompensation, at least all the studies that follow the viral load of the virus, the quantitative PCR studies, the virus is on its way down. And at this point, the immune system starts to ramp up. And it's this maladaptive, the cytokine signature to me looks like a Th17 type response as opposed to a Th1, which would be the T helper cell type 1 response that is better for viruses. So this is an odd cytokine signature for a viral illness. And this is initiated by an interleukin-1, so an IL-1 initiation, but then it goes down this IL-6 pathway. So if you just block IL-6, it's great, but then you end up with the surge behind there. And so my idea of the steroids here is the incredible amount of inflammation you see in the trachea of these individuals. The steroids calms that down. And so I think it's a broad calming of the immune system, but then the IL-6 does sort of more of a surgical strike at the particular arm that is the most maladaptive. So steroids as a broad anti-inflammatory, and then the IL-6 inhibition to try to block the particular arm. And I'll keep talking here because also I'm curious what will happen when people block they go after the IL-1 with things like anakinra. I sort of feel like that might just be more of a sledgehammer to the immune system and not as selective as a modulation. But the big thing that you see in these patients, if they don't get steroids, is if they do end up intubated, if you do try to extubate them, the amount of tracheal and lung inflammation is just dramatic.
All right, Daniel. Thanks so much.
I just want to know, I'll ask Alan's question. How are you, Daniel?
So, I feel actually a little bit better, right? So we're still accelerating, but I have to admit this ability, what would have happened before that was a little demoralizing is, my list every day of, okay, this person's now on 100% oxygen and they're tiring and they're going to end up being on a ventilator for 2 to 3 weeks, if not longer, if they don't get sicker. That was pretty demoralizing. And so to suddenly see that, hey, we can assess and that certain patients, if we start the steroids at the right time, if we do the tocilizumab at the right time in the right patient, just to see something that was clinically so impactful on the natural history we've been experiencing, you don't even need a statistician to show you the difference. Has been, for me, very encouraging, sort of giving me some light at the end of the tunnel that I hope is not a freight train coming my way. And now we're starting to get into a few more sort of the subtleties. Probably people are curious about the Irishman. I'll sort of give you an update on him before I leave. He was the gentleman who, for everyone, would have said that he was going to die. And here we are, day 23 of illness. He's actually still doing well. We were talking yesterday about keeping him sedated because he's a big guy, and he's starting to do well enough that maybe he'll get off the ventilator here soon. But he's getting a point where now he just had a new fever. So now we're going to deal with probably what looks like a secondary infection. So we're getting to the stage of the illness where sometimes people will come in with a primary issue, we'll settle that out, and then they'll end up with a secondary infection. And in the Chinese experience, it would be either secondary bacterial infections or sometimes secondary fungal infections. So aspergillus, things like that. So we're so I am, as much as we're accelerating, as much as the volume, it's just to feel like maybe there is something that we're doing that's making a difference, is heartening. So
Thank you, Daniel.
Thank you, Daniel.
all right, everyone.
Thanks.
Yeah,
See you next week.
Everyone take care.
Thank you, Daniel.
Bye.
Thank you.
One thing that Daniel mentioned is actually on our list today to chat briefly about, and this is a cool paper that came out of Germany, Virological Assessment of Hospitalized Patients with COVID 2019. And this is Berlin, Munich, number of authors, and I recognize Christian Drosten, among others there. And what they did is they did a virological study of patients, limited number of patients, but I think it's 9 patients, if I remember correctly.
9, yes.
They find pharyngeal virus shedding very high during the first week of symptoms. Infectious virus readily isolated from throat and lung-derived samples, but not from stool. But you can find high RNA in stool. Blood and urine never yielded virus. And shedding of viral RNA from sputum outlasted the end of symptoms. Seroconversion occurred after 7 days in half of the patients, 14 days in all. Boy, that's textbook immune response, right? 7 to 14 days.
That's right.
Yeah.
But was not followed by a rapid decline in viral load. So this is a very nice study. They do point out, however, all the cases were mild. And so they suggest that maybe if they were more serious, they would shed longer and there would be virus detectable in the stool as well. But they do agree that there's most likely replication in the gastrointestinal tract. That's why they find RNA in the stool, but they don't find virus. And so,
which I thought was very interesting. I also thought it was interesting, granted these are mild cases, but they're all positive. According to the description in one of the tables, there's fever in only 4 of the 9. Because I always, going into this, I was under the impression that fever was an important indicator, but that I'm getting the impression that you can have this disease without fever.
It also say no virus isolates were obtained from any sample taken after day 8, in spite of ongoing RNA detected by PCR.
That's very interesting too.
It is. I know.
So it shows you the difference between RNA and virus. They
also say that, so basically, they say if these were more severe, we would expect to see higher virus and longer shedding of infectious virus. They also noted that 4 of the 9 patients had loss of taste and olfactory sensation and described this loss to be stronger and more long-lasting than in common cold diseases. Yeah. If you had a cold, I've had a cold where I lost my sense of taste. It's weird. It's very weird. Because you can't eat food. It doesn't taste like anything. Right.
I could probably eat food if it doesn't taste like anything. I'm pretty good at food.
Yes, especially like right before a TWiV, you have no problem stuffing it in.
You bet.
This study has some bearing on when you release a patient after they think they've been cured.
Well, as Daniel said, they still have symptoms when they're released. You can't.
Well, I mean, even if they didn't, they're still shedding virus.
Yeah.
Yeah, for a couple of days after symptoms. Yes.
So maybe there should be a 3-day or 5-day, self-quarantine after you're released and you feel great. You feel like going out and running a mile. You should actually go back home and let that feeling go away for a while.
Well, that was the recommendation. When your symptoms are gone, wait at least 3 days,
yes.
Which sounds to me, and I think that's what I understood Daniel to say is that that's the current CDC recommendation, is wait 3 days after the symptoms are gone. And from what I'm thinking is that that's kind of a minimum. Okay, I'd want to hang out for maybe a week if I could do it.
Right.
Maybe 2 to be absolutely sure.
Right. That's what he said. If you're retired and you don't have to go anywhere, maybe just stay another 14 days.
You could be happy that you recovered, right?
Yeah.
Exactly. Yeah.
Another paper I wanted to mention, it's a bioRxiv preprint from the Harbin Veterinary Research Institute and the High Containment Laboratory for Animal Disease Control, also in Harbin, in China. Susceptibility of ferrets, cats, dogs, and different domestic animals to SARS-CoV-2. So they looked at these animals and especially ferrets and other animals that would be in close contact with humans, right? I guess all these could be in close contact with humans. Anyway, they found that the virus replicates poorly in dogs, pigs, chickens, and ducks, but pretty well in ferrets and cats, and the virus transmits among cats by respiratory droplets.
Did they look at transmission from humans to cats?
No, they did not.
No.
Okay.
Likewise, obviously not the other way either.
That would be a tough experiment, right?
Yeah, but this goes back to the discussion with Ralph about the potential for spillback,
yeah, for sure,
from humans into an animal reservoir, and that being a reservoir, and this is a particularly, shall we say, interesting situation if the reservoir was an animal commonly found in houses as a pet.
Well, the good news is that dogs are not easily infected, but cats seem to be. So what would our recommendation be if you have COVID-19 and you have a cat?
Huh.
People are going to ask, you know it.
Yeah. Yeah.
I would say don't kiss your cat while you're sick. People do that.
And keep the cat indoors.
Well,
if that's possible.
Yeah, cats tend to wander around the house, right? It would be good if you could put them in a separate room from where you are.
Mm-hmm.
I'm just thinking, we go out walking and there's cats around our neighborhood, and we always go kitty, kitty, kitty and go pet the cat, right?
Yes. Mm. So I think you should be careful with cats and ferrets. People have ferrets also as pets, right?
Yeah,
right.
Saw one on the street the other day.
Mm. Oh, when we went to see the bats in Austin, someone was there with a pet ferret. I remember that.
Yep.
So they say something in the paper which I think is interesting. They say it was difficult to perform regular nasal wash collection on the cats because they were aggressive. So they only collected feces.
Yeah, I've seen joke cartoons about the difference between giving a pill to a dog and giving a pill to a cat. Okay, pill for a dog, you just stick it in their food and they eat it.
Yeah.
Trying to give a pill to a cat is a major project.
Oh, so that's why Alan just texted me. He can't join us because he's trying to get a pill into a cat.
Is that right?
That's right.
Okay.
That's great.
Oh, I didn't know it was so hard. Yeah, because we just put it in food for the dogs. Anyway, so this doesn't mean that every cat is going to be infectable, right? Cats are different, just like people, but you probably should be careful until we have more information. Is that reasonable?
I think, in a way, the most significant part of this study is that the cats could transmit it from one to another via aerosol. Now, it was, I think, it's a limited study. They had like 3 cats in a group. It was one of the 3 cats transmitted the virus. So, it can happen. That doesn't mean it's always going to happen, but it's worth thinking about.
Yeah, of course, in this experiment, they put the virus right in the nose of the cat, right, with a pipetter. So it's not going to happen in real life. And the dose is probably higher. A couple of other interesting tidbits here. As you heard from Daniel, the New York State is recommending wearing cloth masks in public. And the U.S. is also expected to advise everyone to wear cloth masks, according to the New York Times today. And this is an interesting turnaround, right? The CDC has been so far saying no, no, no. And now it's I'm not sure I think it's a great idea to wear masks as long as Daniel said, you wear them properly. But I'm not sure. I guess it's the matter of so many infections now. This is the right time to wear it. What do we think?
Well, and this recommendation is for at least cloth masks. It's not saying N95 masks.
Yeah.
And there are hundreds, if not thousands, of people sewing masks at home because that's something that they can do. Yeah. And so, in fact, there's this paper that Alan sent us about from Nature Medicine, respiratory virus shedding in exhaled breath and efficacy of face masks. So there's a study of 246 individuals with seasonal respiratory virus infections who were randomized to wear or not wear a surgical face mask, again, a cloth mask. And the results showed that the masks could significantly reduce detection of coronavirus and influenza virus in exhaled breath and may help interrupt virus transmission.
That goes back to what we've talked about before, in that one of the primary uses of a mask in the everyday situation is to prevent you from spreading disease if you happen to be ill. Okay. Mm-hmm. And that, I don't think we've ever debated that. And the link that you put into that blog had up front a pretty good summary of the situation with face masks and what we know. So I think the notion that it's good for preventing spread from someone who is sick to other individuals if used properly. I think we've all accepted that from the beginning. The biggest question has been whether a surgical mask, even worn properly, is effective in reducing the chance that you are going to get infected from somebody else. And there's a lot of debate on this, but, there are other reasons for wearing a mask, in particular, since there is pre-symptomatic or asymptomatic shed. So you can have people walking around who don't know they're sick, who might otherwise spread the disease. And then I think going back to an argument that Vincent and others have made very many times, and that is it keeps you from touching your face. So there are good reasons to do it.
So this study, Kathy, they actually had sick people and they had them breathe through the mask, right? And then they measured what was, so that's very interesting. And in fact, the graphs are quite striking here. Mm-hmm. But they don't actually look at illness, though. They just look at virus passing through the mask, right?
Right. And I would go back to also, I think it was Ian Lipkin who said, basically the way everybody should be thinking now is that they themselves are infected, but maybe not yet symptomatic, and assume that everybody else is infected and maybe not yet symptomatic. And given that, wearing masks could help flatten the curve, kind of as Daniel was saying. Again, remembering that it don't touch your eyes either, and I was thinking, if I'm going to go out, I'll be sure to be wearing my glasses as well as, a mask. If I go to the grocery store, maybe a cloth mask.
Yep.
There's all kinds of patterns. I think you had links in previous shows, and Vanderbilt has a pattern that they like and so forth. So if people want to sew up something, you can.
So, there was my wife discovered a kind of a podcast, a video of a physician in New York, working at Cornell, talking about the disease. And he was talking about the use of masks. And his whole rap was, as we've said all along, that a major vector is from your hands to your face, and from your face to your hands and to other items. Mm-hmm. And so, wearing a mask helps cut down on the spread if you're sick, but it also helps keep you from touching your face. So washing your hands isn't, after you go to the store is imperative. Okay. But if you're not really careful while you're in the store, you're going to touch your face. And so wearing a mask is going to prevent you from doing that. So, I think there are good reasons for doing this.
I think we should also say we've changed our tune in the course of the last 3 months. Okay.
Yeah.
We certainly have.
Yeah.
Mm-hmm.
Because initially we didn't have all that much data. Now there's more. And personally, yes, I would wear a mask should I go. I went to the post office the other day. I wore a mask. In fact, at the post office now they have plastic sheets in front of the counter and they're all wearing masks, which is the right thing to do. So I'm happy to admit that we were wrong in the beginning.
Yeah. There's a letter that addresses this later on. I think the important thing is you can be wrong. And the important thing is to say I was wrong and adjust to the situation. It's not a matter of being wrong it's not important. It's a matter of doing the best you can in the moment. Okay. And trying to be responsible. And part of being responsible is saying, things have changed. My perspective has changed.
Yep.
My wife, Marlene, has made masks for all of us. And she got the brilliant idea of going to the vacuum cleaner and getting the piece of HEPA filter and putting that between the two pieces of cloth to give an extra barrier. And we've inserted a small wire so you can squeeze it up against your nose. And they were quite effective, actually. Mm-hmm. So it's like an arts and crafts project with a meaning.
Yeah, one of the patterns I saw had the wire you could use, even like the twisty ties that come with bread or something like that.
Yes, my wife is using pipe cleaners for the nose part.
Mm-hmm. Mm-hmm.
This is going to become
Absolutely.
a fashion statement, right?
Mm-hmm. Yeah, of course.
I'd like one with a smiley face on it.
I want a virus. I want a coronavirus on mine. Is that like morbid? Yes, go ahead. Call me morbid. 2 more links I want to tell you about. First, there's an article. Japan is going to give cloth face masks to 50 million households.
Wow.
That's great. And then I found this blog post, which I think Rich mentioned, with all you ever wanted to know about face masks. And the author does a good job of sorting it all out and trying to figure out why the CDC said for so long not to wear them. So it's worth a look. And I think Kathy has a few things to tell us, right?
Oh, yes. So, well, there's the Where America Didn't Stay Home Even as the Virus Spread. Someone else put that in the big map from the New York Times. It's on the front page today above the fold. And just showing the states that...
Yeah, that's very disturbing, isn't it?
Yeah, the states that have already had stay-at-home mandates and from when they had them and where people are traveling based on cell phone things. And I can't remember where it is, where now, maybe it's Seattle, the average distance that a cell phone moves is only 61 feet. And yeah, so it's quite impressive.
But there's still a lot of travel. That's normal in this country, right?
Right.
And it looks like in Austin, Texas, they're normal. I don't know where Austin is. I'm just kidding.
No, Austin looks like they've cut down their movement.
Where's this link? I'm missing this link.
Oh, it's this dark black Where America Didn't Stay Home. It's actually a link.
Oh, okay.
Yeah. Tell us if Austin is white or red, Rich Condit.
Let me zoom in here.
Yeah, it's a different coloration from what's on the front page of the New York Times, but
Kathy,
same map.
they cite a University of Michigan Professor Aaron King. Do you know him?
No, but I did read about him in some Michigan thing that we got, I think. I don't know him. Yeah.
So I just put this up because I think it's too bad that many people in the country, maybe half the country, they're still moving around as usual.
It's true.
It's not what we're supposed to do, right?
Yeah, Georgia and Florida now have stay-at-home, but at least I think it's in Florida that the governor didn't want to forbid gathering in churches. Mm-hmm. No, I don't think it's. You think? Well, I don't know. I don't remember which state. It might be more than one, but they're still allowing congregation of more than 10 people. So that's not a good thing.
The admission of the Georgia governor to not know that there are a lot of people out there that are asymptomatic walking around was just ludicrous.
Yeah, I didn't see that.
I can't really localize Austin specifically on this, but my sense from looking around the area is that it's less than normal travel. And the Texas governor has not issued a shelter-in-place order. It's some sort of less stringent recommendations, but
okay.
So then I wanted to point out on Virology blog, which is Vincent's blog home, he sometimes has visiting bloggers, and so Trudy Ray's post went up, and it presents hypotheses for why COVID-19 is less severe in children, and I think it's a good read. She's put in a number of good references, and then one person added a comment of another possible contributing factor. And then it turns out that the following blog, which is from a ME/CFS patient, so that's myalgic encephalomyelitis, formerly known as chronic fatigue syndrome. So this is a patient for whom stay-at-home world has always been, or recently has been the case for her, and describing what it's like. And she's been doing grocery delivery for years, and now it's hard for her to get grocery delivery because everybody else is doing it. So
she's been doing it for 13 years. Yeah.
So another thing I found was a follow-up. It was in the U of M Daily Online News. Krista Wigington is involved in a sewage study with collaborators at Stanford regarding whether a sewage surveillance effort would help track COVID-19. And so her specialty, or at least the way I know of her, is her work on figuring out how to inactivate viruses in water. So that's going to be interesting. And then we have a letter from a listener, Greg, who cites a Lancet article where they've already tested water from the Schiphol airport in Amsterdam, looking at the same thing. So it looks like this is going to be a feasible thing. I think it was last week we were talking about that.
Right. And it looks like they started surveilling when there wasn't much disease and it was negative, and now the sewers are showing up positive. So that's very interesting. Astute question from a listener.
Mm-hmm. Then I put in a link for National Geographic coverage. All of their coverage is open access, and I don't know how it happened, but I am now on the mailing list, and I'm getting emails virtually one a day from them about something. And because I'm teaching influenza on Monday, mostly the epidemiology and history, it really struck me because it's an article about how in 1918, social distancing was employed to various extents in different United States cities, and which cities peaked relative to when the social distancing happened, and some recovered, whereas others recovered but then rebounded, and so forth. And so I thought that was interesting. It could have some lessons for us for the future. And it's a 3-minute read and really nice graphs and things.
And the bottom line was basically right that the social distancing has an impact, both on the on the number of deaths per capita in the end, and the impact of the disease generally. I found this fascinating. This is great. Nice graphs.
Yeah. Yeah. And really timely for my course. So I'm happy about that. And then another thing I put in, but I'm trying to remember if I already heard you talk about this, where you can do citizen science. And even if you're healthy, report how you're feeling to covidnearyou.org. And takes about 15 seconds. And then it's helping them get a denominator. And then if you start to have, maybe a sore throat or runny nose, then you might say, oh, I'm not feeling as well today. And then you get a little bit different set of questions, but still very short. And they have a map here of all the people who've reported symptoms. And then if they followed up and got tested. And so they're trying to put that data together.
I think the name is interesting. COVID near you.
Well, they've had a flu near you study for over 10 years. So it's building on that same kind of thing.
Vincent, have you heard what they're calling people that disregard all of this advice? What? They're called COVIDiots.
Oh, yeah. I buy that.
Oh, can I mention this polio story?
Yeah.
Of course.
So Alan pointed out on our Slack channel that the SARS-CoV-2 pandemic is forcing polio eradication groups to halt their campaigns.
For 6 months.
In many countries, this is ongoing because we're not quite eradicated. Afghanistan and Pakistan still have circulating wild virus, and they have to immunize there, and they can't do it because you don't want to spread virus inadvertently from household to household. So they've had to stop. And I just wonder, it's going to be for months, obviously. I wonder what the effect will be if, we'll see an explosion in the number of cases in those countries and start to spread elsewhere. That would be unfortunate, wouldn't it?
Mm-hmm.
Very much so.
Wow. I hadn't thought of that. It's a good point.
One thing that's a follow-up from, I think it was TWiV 596, when Alan in particular was talking about healthcare workers not going home, but staying away to protect their families. In both University of Michigan, Ann Arbor and Flint campuses are going to be offering dormitories for healthcare workers at $20 a night. And Alan brought up the point of it, separating these healthcare workers from their families at a very difficult time. And when I was listening, I was thinking about how military families do this, particularly in wartime, but also in peacetime, to which he counters, that's true, but healthcare workers' families didn't sign up for that. And I would argue that in some cases, the military's extended family also didn't sign up for it. And yet, if the military person feels compelled to join the military and go to war, and the healthcare worker signed up, and, especially these ones that are, coming out of retirement and so forth, that maybe, staying in the dorms, if that's what they think is going to be good for them and for their family, it's nice to have that option, I think.
Yeah, it should be up to them.
So I did want to ask Vincent, at the very last 6 or 7 minutes of the 596 that you, Alan, and Rich did, you talked about the mechanism of the hydroxyquinoline resistance and also the azithromycin. Is there a potential for SARS coronavirus to develop resistance to either of these, do you think?
I think hydroxychloroquine,
it's the right thing. Yeah.
We always say there's no drug that a virus can't become resistant to. So the thing with hydroxychloroquine is that it probably has multiple mechanisms, and one of them is elevation of endosomal pH. And you can imagine that the spike would sustain amino acid changes that would change the pH needed for fusion, right? So,
okay,
one reaction is that the potential for resistance might be less because of the fact that it's a drug that targets the cells rather than the virus. So, it seems like it's not like you can come up with a virus mutation that no longer binds the drug or something like that. The virus has to come up with something that circumvents what's going on in the cells. On the other hand, although we're talking completely different organisms, I am correct in saying, right, that malaria develops quinine resistance, right?
They also develop chloroquine resistance.
Okay. So there you go.
So the thing is that I think chloroquine probably has more than one mechanism of inhibition, right? So we don't really know. The endosomal pH is an obvious one for viruses that have to get in via endosomes, but chloroquine has a lot of other effects, and no one's sorted out which one is most important for SARS-CoV-2. So, if there are different ways that it's inhibiting, it might be harder to get resistance. But yeah, it's an interesting issue. I looked this morning after you put this link in, couldn't find any papers describing chloroquine resistance to viruses. Now, I didn't do an exhaustive search, but, I go through 10 pages of PubMed, and then if I don't find what I'm looking for, I give up.
Yeah.
But there may be there. Anything else before we go to email? Oh, yes.
Just
the patch.
In this thing from the NIAID, direct email from NIH that I got, prickly patch delivery of experimental COVID-19 vaccine shows promise in an animal study. So this is scientists at University of Pittsburgh tested this microneedle thumbnail-sized patch, and they tested it on mice, and they published it on April 2nd in a journal, Lancet journal that's called eBiomedicine. And basically, this is the popular press type of article from the NIH, and it seems to generate a surge of antibodies against SARS coronavirus 2 within 2 weeks of the microneedle prick. And the animals haven't been tracked long term, but they noted that mice vaccinated with microneedle patch against MERS produced sufficient antibody levels to neutralize the virus for at least a year. So far, they're following a similar trend.
This goes back, we've discussed these microneedle patches in the past, and it goes back to some of those discussions and some papers that we've talked about introducing antigens right into the skin. So this has always been a really interesting technology to me. One, because it gets around actually having to inject somebody. This patch would maybe, I doubt if you can actually even feel it as scratchy or anything. It just has a bunch of 400 something microneedles on this patch that apparently dissolves. So you just put it on the skin and let it go away. And I think it would be at most mildly irritating. But more important, it delivers the antigen right into the skin, which is a hotbed of dendritic cells, which are the cells that present antigen. Those are the cells that you want to capture antigen and carry it off to lymph nodes to stimulate B cells. So it strikes me as a really, really interesting technology, and I hope we see more of it.
There's an interesting twist to this, though, because mice are not highly susceptible to this virus, as shown by the animal studies, but cats are. But cats are not allowed to be worked on anymore at the NIH. There was a protest, and they had to get rid of all their cat research. So to find an animal model that will fit this vaccine development, they'll have to change a regulation there, I think.
Weren't ferrets infectable in that paper we just...
ferrets and cats, yep.
Right, but
yeah, you could use ferrets.
Okay, use ferrets.
I think these microneedle patches are going to replace needles one day, and that would be great.
That'd be great.
Because so many vaccines are delivered by needle, and this would no longer need to have a trained healthcare worker.
Yep.
Even I could put a vaccine on someone.
I don't know if I'd let you, Vincent.
Okay, no worries. All right, let's do some email. First one is from Bradley. No, is it? Yes.
Yeah.
Why can't candidate vaccines be manufactured in industrial quantities prior to passing the tests? If they don't pass the tests, dump them. Normally, this would be wasteful, but not in the current crisis. Current with a capital C. Well, that could be done. I am a bit cynical because I think the companies want to make money, and this would affect their bottom line. But perhaps some cynicism is put aside in this crisis with the capital C. However, my wife points out to me that the regulatory requirements for mass production are different from testing. And so you would have to have all that worked out ahead of time. Seems to me that the U.S. government should pay for this, right? And I think that's a great idea, though. What do we think?
I have this vague feeling that this is the anecdote that I thought I read within the last day or so, that Moderna, that's doing this phase 1 clinical trial of their vaccine, was anticipating larger scale production. So that at least they're figuring this out, at least on paper. Okay.
Yeah, that's a problem, right? Which one to large scale? Because there are over 50 different candidates. You can't do them all. That would be crazy, right? I think you have to get at least some data before you would make that decision. By the way, could I take a 5-minute diversion here?
Yeah, sure.
So the Moderna mRNA vaccine, in all the papers you read that have been published, they make this RNA by in vitro transcription.
Correct.
What are they going to do to scale that up? I can't, we do 50 microliter T7 transcription reaction. I can't imagine liters, or maybe that's what they're going to do. They could synthesize it, I guess, but it's a pretty big coding region.
No, you're right. It's a problem. And let me just find an email here, because I had an exchange. Remember, I work on this Brighton collaboration group. Okay.
Right, they pulled you out of retirement.
Yeah. And so I had an exchange with them about the difference in I had an exchange with one of the other scientists about the difference between messenger RNA vaccines and self-amplifying messenger RNA vaccines. The self-amplifying messenger RNA vaccines are based on an alpha virus platform or similar viruses, where it's they've replaced structural proteins with an antigen, and they deliver this as an RNA, which encodes not only the structural proteins but also some replication proteins, so that the original delivery of the messenger RNA, it's a positive strand RNA virus, is ramped up by replication during infection. So that helps you get more dosage, basically. And in talking about this, he said, as a point of reference, to immunize the U.S. population with 3 doses of 250 micrograms of an mRNA vaccine would require 245 kilograms of GMP-compliant RNA, plus 15 to 20% overage. And he says that's not going to happen. So that a dose-sparing capability of self-amplifying RNA vectors is an important factor. So
Moderna is not
a very significant question.
But Moderna's is just RNA; it's not amplifying, right?
That's right. That's what he's saying. Is that if you don't amplify, that's the kind of scale you're going to need. 245 kilos of RNA. Okay, wow. So you're absolutely right. Scaling that up is something. I also thought, how long ago? 1973? What's that? 47 years ago? Probably there were all T7 RNA polymerase purified in only 2 places in the world. One was in Mike Chamberlain's lab in Berkeley, and the other was in Ed's lab at Yale. Okay? How far we've come. T7 RNA polymerase. Amazing.
Dixon, can you take the next one? Bradley.
You did Bradley. I thought you didn't do Bradley? Okay, fine.
No.
Okay, partial answer to question TWiV 596 as to whether different languages create different aerosols and are thus more effective. Aerosol, and then it says there's a big website listed, and then finally it says aerosol emission and super emission during human speech increases with voice loudness, he said softly.
Doesn't matter. Yeah, it doesn't matter what the language is.
It doesn't matter what the language is.
So, Dixon, you would emit viruses the strongest of all of us.
Thank you. I'm a good lecturer. Aren't you glad I'm sitting in Fort Lee?
In this study, one of the things that was interesting in this is that they identified people who were super emitters.
Mm-hmm.
And they couldn't really tell why it was they were super emitters, if I understood it correctly. But they projected a lot more particles than other individuals. Interesting, given that there are super spreaders.
Right. Right.
Well, thank you for that. We appreciate it.
Mm-hmm.
Kathy.
I'm clicking back. Magda writes, Dear Microbes, I was wondering what you think about disinfecting our personal phones after every use. Is there any point in washing our hands more often if we don't disinfect our personal phones? I'm a veterinary surgeon from Poland working in the UK. And I think that probably it's not practical after every use, but periodic disinfection is probably good. So for myself, I tend to, when I've been out, just outside my house, I haven't been to the grocery store yet since the stay-at-home order, but I tend to wash my hands and then I wash my phone. And then I wash my phone quite a bit more often than I had been. So I think it's a good point.
It's a very good point. And Magda loves our show and now wants to be a microbiologist.
Excellent.
Excellent.
Maybe after there's going to be a surge in virology, microbiology careers. That would be cool, right?
Right. We think we're getting a lot more take rate on our graduate admissions for our doctoral program in microbiology. Yes.
Cool.
That we're not sure if that's why, but yeah.
Oh, when the economy goes down, that's
people
what
go to graduate
school. Yes. Rich, can you take the next one?
Russell writes, having tracked this virus with you from TWiV 585 or thereabouts, I have always noticed a calm demeanor when approaching it, almost as if it were just another run-of-the-mill virus. As the shows went on, I could see that it was more serious than we all believed at first. Any reflections on if TWiV underestimated the virus and its impact and what we could have done differently if we all understood the severity of the Wuhan situation? Well, yeah, I have comments on that. I certainly underestimated the virus and its impact. And I've thought a lot about this because I don't feel great about it, but what the heck? Like I said, closer to the top of the show, sometimes you're wrong. I think one of the reasons is denial. Like, this can't happen to me. Plus the lack of a precedent. The last time anything like this happened was 100 years ago. And so I had trouble, just accepting the notion that it could happen. And I think this whole thing is a teacher. And in terms of what we could have, what this will impact our thinking in the future, having seen this happen, I would hope that there would be a lot more preparedness, both intellectually and boots on the ground. And what we could have done differently, I think the U.S. could have, the U.S. and other places probably could have taken precautions earlier. And I would hope that in the future, there is a pandemic preparedness plan in place so that when something like this hits, we don't have issues with communication, with understanding how to respond and getting the population to cooperate in responding, with having an appropriate supply of medical equipment and et cetera, that we would be ready to go and deal with it from the beginning. This is a great teacher.
But, Rich, there's a curve of forgetfulness. And so that's it. 1918 to 2020, that's the curve.
Yep. Absolutely.
Back in January 17th, my first session of virology class, I said to the class, 120 students, who's heard of this new coronavirus circulating? And 3 people raised their hand.
That's the same proportion as in my class, which was, I think it was around January 13th. It was 1 out of 42 had heard of it. I would agree with Rich. I think we did the best we could at the time. And I feel a little blindsided in retrospect should have thought more about coronavirus. I think all my life I've thought, okay, when the next pandemic comes, it's going to be influenza. And, I tended to think of it through those glasses, and the idea that, well, we have thousands of people getting influenza every year, and yet people are complacent and don't get the vaccine, and was just trying to think of it along those lines. But, as the listener points out, as time went on, we started to see how much more serious it was.
The other fallacy that I maintained was that I would do the comparison with flu as well. And I would say, well, in this country, we have 15 million cases of flu every year and on the order of 37,000 deaths and many more hospitalizations, and we can deal with that. But what I didn't realize that's becoming apparent now is that that's part of the background. And the health system operates very close to capacity with that as background. Mm-hmm. And if you add the pandemic on top of that, all of a sudden you got a problem. That's where the preparedness comes in, is to have some program in place to be able to handle the excess morbidity and mortality, the excess hospitalizations in an event like this.
Our hope is also that the coronavirus vaccine works a lot better than the influenza vaccine.
Yeah, I would go. I'm with you on that one, Dixon.
Yeah, I keep thinking of this in terms of the idea of the error threshold for RNA viruses, and RNA viruses are, very close to the error threshold, and we're at the very threshold of capacity, like you were just saying, Rich. And so we don't have that stretch capacity that we need for pandemic preparedness.
Actually, the other pandemic preparedness thing I like is what I will call the CEPI model, which is to have a publicly funded program for researching and testing vaccines for potential pandemic pathogens, up all the way through a phase 2 trial, and with a stockpile of enough vaccine to initiate a phase 3 trial when that comes around. Now, of course, the problem there is identifying what potential pathogens are, but some thought in that direction, I think, is important.
Well, WHO has a list of these pathogens, and those are that's what CEPI works from, and it includes Nipah and so forth. And the last virus on the last pathogen is pathogen X, the one we don't know about yet, and that's what we are now at pathogen X.
Yep.
Dixon, what did you think in the beginning of this all? Did you think it was not going anywhere?
Well, I'm not a virologist, so I get to opt out of this opinion because I made the biggest mistake about the H1N1 when it came up out of Mexico. So I remember what I said, much ado about nothing, and I won't say anything anymore because there's no way to predict this. There's absolutely no way to predict it. And
that was the title of a TWiV, much ado about nothing.
So you expect the worst and hope for the best, and that's what we should have done. And
I thought this was going to be SARS-like, SARS-1, which fizzled out in a year.
Harder to transmit, that's true.
Yeah, but I didn't realize it would be a better transmitter. Thank you. That was very thought-provoking, Russell. Thank you very much. All right, Yossi writes, Yossi is a med student in California, and like many other med schools, they're graduating early, and so they can go to work. Mm-hmm. Very cool. I hope you don't mind. I took the liberty of quoting your podcast in a policy brief I wrote. I'd like to convince more lawmakers and funders to prioritize developing drugs, especially antivirals, in advance of pandemics, and we have a link to that. My question, what are your thoughts on a future where we have a roster of drugs against all known viral polymerases, for example, or some other suite of drugs specifically for pandemic preparedness? Well, I think this is a great idea. Whether it's feasible or not, we don't know, but we'd have to try, and no one has tried to do such a thing. But I argue, and she quotes me in this article, that we could have been ready with antivirals against coronavirus polymerase, for example, but no one was was doing that. So I personally think, I hope this pandemic is a motivator, but I also am very cynical. And as someone just said a few minutes ago, the forgetfulness will set in very quickly, I'm afraid.
As time goes on, there's going to be an accumulation of drugs that have antiviral activities for some targets in common. And I'm thinking about how a lot of the HIV drugs that were developed have activity against hepatitis B, and I think even some against hepatitis C. So that as time goes on, I think we're going to have a larger and larger selection of drugs that, when a new pathogen comes along, are already approved and you can screen against them. But I would wholly support an effort to expand that panel further.
Just think if this $1 trillion or $2 trillion or $3 trillion had been, some of it been put into some basic research, we wouldn't have had to spend that.
Right. This policy paper that Yasi has written and provided for us, I think it would be a good thing for people to take a look at. Headers like just-in-time drugs are too late, and calls for in-advance therapeutics.
Yeah,
it's very well written.
So I think, it's definitely for anybody who wants to think about the economics of drug development and take a look.
All right, the next one is for Dixon.
Okay, here we go. Cheryl writes, I am an attorney/broker by profession, but I have a passion for biology and natural history. Keep up the great work. We are listening. Then she writes, hopefully the current viral pandemic will be a wake-up call to governments to make it a top priority to incentivize and fund scientific research in virology so that we can be as prepared as possible for future pandemics. And that was what we just talked about. But I must say that some people will argue, well, it costs as much to do that work as it does to make a nuclear aircraft carrier. Speaking of which, there's an aircraft carrier that's nuclear that's sitting off the coast of, I believe it's Guam, which is inoperable right now because of the pandemic that's sweeping through that ship. So if we had this available to us before, we could avoid the clash between the Navy brass as to what's going on there. So I think all kinds of interesting sidebars all come together when we're not prepared.
You know what's interesting? It could be that the military takes the lead.
Exactly.
In the next round of drug development, because they'll see this incident and say, we can't handle this.
Look at Vincent. What if 90% of the crew is offloaded from that ship? They can't operate.
No, of course not.
They've got 60 airplanes. They've got to launch all those. They can't do that. It's just it's horrible.
The military is why we have an Ebola virus vaccine, because, 20 years ago, they started the development because they wanted to protect troops. And so this is very interesting. Cheryl, by the way, works in Hawaii. All right. Kathy.
Yuri writes, I'm just a human being who loves biology in general and virology in particular. I'm a graphic designer and I don't have many people to talk to about biology, so you are all my virtual friends and guides. Last time on TWiV, you talked about low probability of antigen drift in SARS coronavirus 2. At the same time, I've heard from several virologists that it's a common thing among coronaviruses. I've looked up and I found an article about gene drift of the spike protein gene in several strains of human coronavirus OC43 and another about H coronavirus 229E. So these are what we call the common cold coronaviruses. Why don't you think that SARS coronavirus 2 in the future won't evolve in several strains? Does it connect with the genome structure somehow? I mean that influenza has a structured genome, and I think by that they mean segmented genome, and coronaviruses have a single RNA strand. Or is there something else? I'd be pleased if you could shed light on my question, because I think it intrigues many people. And so Vincent's put a link in here to listen to TWiV 597 with Jonathan Udell, which was just released this morning. So I hope listeners will forgive us for not knowing what's in that podcast yet.
Yeah.
But the gist the gist of it is that, everything with a genome exhibits some genetic drift, even DNA viruses. Coronaviruses are RNA viruses that have a proofreading enzyme, and so they have a drift that's maybe slower than that compared to other RNA viruses. And so why don't we think that it's going to evolve in several strains? I think the evidence so far is that the virus is pretty optimal. And what we know about virus evolution is that it tends to, the RNA changes that happen tend to make the virus less virulent for the host. In other words, to evolve towards something that's going to allow it to survive longer in the host. But there can be evolution or selection for something that can transmit better.
I think Jonathan Udell put it really well. So I encourage Yuri to take a listen to that. But basically, the drift is genetic, as you say. It's not antigenic drift among the coronaviruses. It's just not seen. So there's no reason to. He has a wonderful argument for this, and I will leave it to listen to him because he puts it really well. Wouldn't you say, Rich? He did it really well.
Yeah. However, I do want to. I haven't looked at these two links that Yuri sent in, and they look interesting. So maybe we can put them aside and have a closer look at them, and maybe even come back to it another time. Has anybody else looked at those?
Yeah, they're genetic drift. It's not antigenic drift. Yeah, it's nothing that would affect the antibody interactions. Yeah.
Okay.
And I don't think it relates particularly to segmented genome versus monopartite genome.
Agreed.
It's an interesting hypothesis, but
agreed. One other thing that's very interesting. So I did a TWiV with Nels LD yesterday. Should go up tomorrow. And I asked him to explain the nextstrain.org, which is a site where you can go and look at all the genome sequences of SARS-CoV-2 that have been accumulated so far. And they put them on a phylogenetic tree, and you can see what country they came from. And he just pointed this out, which I didn't realize. From the first isolate to the current, there's a total of 15 base differences.
Whoa.
15.
That's all. Yeah.
That's it. And people he said, don't think of this as like a tree of life with cows and dogs and people were separated by millions of years. This is 15 bases. And that's a perfect way to look at it, right?
And a lot of those are silent. Is that right?
Yeah. Many of them are silent. They're just things that happen, right? But, we have so much data that it looks like, and you have all these branches, right? It's, oh my gosh, it's this branch and this, but they're really similar.
It's interesting because that's remarkably stable.
It is. It is remarkably stable.
Probably among other things because, just doesn't, maybe you can probably get a lot of, and I think Jonathan talked about this, you can get a lot of variation, but in particular, since you go through a bottleneck every time you infect somebody, everything's got to go right. There's not much that it, in the end, doesn't tolerate a lot of that variation.
And you can calculate from the genomes that have been sequenced that this virus is changing at a rate of 26 nucleotides per year. Okay.
Not much.
That's not a lot for an RNA virus. Rich Condit.
Anonymous writes, why is everyone urging more testing? If your symptoms are not severe enough to go to the hospital, you stay home and heal on your own without spreading to other people. Why does it make a difference by testing more people without symptoms or mild symptoms? That was one question. The other is, what's your opinion about Japanese flu drug Avigan? It seems very promising. Testing just, I think, helps us to manage the pandemic better if you know how many people are positive and where they are. The more data that you can get helps you understand what's actually going on in the population. Any other thoughts on that?
Yeah, I think you need to go beyond the sick people. We have no idea right now how many people are infected in the U.S. And if say half the population were infected right now, it would really make a difference in our predictions, right?
Right. I've thought that there's been a lot of modeling about what's going to happen, what the curves look like. And I'm sure that, the people in government in particular are struggling with how much restriction you put on or you suggest or mandate balanced with the economic impact. I'd like to see a model that was a collaboration between an epidemiologist and an economist, okay, that looked for, this is actually, I suppose, could be thought of as cynical, but, people suffer serious economic consequences from this. And it would be interesting to have some sort of information, some sort of forecast of the economic damage relative to the damage just from the disease itself. Testing would be part of that. The more testing we have, the better you can make those predictions.
Yeah, it doesn't hurt to mention the fact that the village in northern Italy, Vo, tested everybody and found 89 infections. Those were all asymptomatic people. They waited 2 more weeks and they tested everybody again, and they only had like 3 infections. So it helps you in controlling infections on a very small level, but you could multiply that out.
Yep. By the way, favipiravir is an antiviral that has been licensed against influenza in some countries, including Japan. And it's not going to be licensed in the U.S. It's a nucleoside analog. However, it is in clinical trial for SARS-CoV-2. So we'll see what happens.
What's the mechanism of that?
Nucleoside analog.
Okay, so that would be a target replication.
Yep. RNA polymerase.
Okay.
Sanjana, junior undergrad at Caltech, currently at home in South Bend, Indiana, aspiring virologist or immunologist. Estimates for the basic reproductive numbers are about 3 for SARS-CoV and 2.2 for SARS-CoV-2, according to this study linked. But it seems to me that intuitively SARS-CoV-2 is more contagious because the former has reached pandemic level, though perhaps SARS-CoV-2 could make up for a lower R0 with a large asymptomatic and pre-symptomatic transmission. With confidence intervals, though, I'm not sure I'd believe that there's a significant difference between the two virus R0. So how do you measure contagiousness or infectivity as an intrinsic property? Well, my understanding is that you need to do this early in an epidemic before there are interventions, because interventions can influence the transmissibility, and then it's no longer an R0, which is the basic reproduction number, but it becomes an R, which reflects interventions, right?
Right. Or an RE, effective reproductive rate.
So this one was identified early on in China in a very early outbreak, and I think that's going to be the R0 that we're stuck with. And if you look at SARS-1, it's higher, but remember, most of the infections happened in hospitals, and so that's kind of an artificial situation, right? We put people, before we knew how to do infection control in hospitals, especially. So I think that's higher than an actual R0, but it wasn't done because, remember, we didn't hear about SARS-1 until months into the outbreak. And so by then it was probably too late to calculate the real R0. That's what I think.
It looks like this paper, in fact, is an analysis based on, it's titled Early Transmission Dynamics in Wuhan. So it's doing exactly what you suggest needs to be done.
That's what we covered that months ago. Yeah.
Oh, okay.
Yep.
So, and the other thing I would say is there are many other reasons that classic SARS was we were able to control that better than SARS-CoV-2. And I would go back to our episodes with Ralph Barrick, where that's been discussed in some detail. Yep. It's not just the reproductive rate. Other characteristics of classic SARS enabled control of that more effectively.
Dixon, can you take the next one?
Yes, I can. Kevin writes, on a recent episode, you discussed testing sewage for SARS-CoV-2 RNA as a public health surveillance measure. Apparently, this is already being tried in the Netherlands. And he gives a reference for that. Cheers. He's the Associate Director of Environmental Health Sciences and Compliance at the New York Genome Center.
Yeah, first detection in sewage. That's just so interesting. We talked about this earlier. Someone asked about it, and the study comes out.
It's great.
Mm-hmm. Mm-hmm. Yeah, I didn't find this one. Yeah, I didn't find this one when I was talking about Krista Wigginton's work, but a little bit further down, we have the same link from Greg. Or actually, this one is a Med archive, and the link from Greg is for something that's been published, but could likely be the same story.
Can you take the next one, Kathy?
Sure. Janet writes, I'm trying to find the question. Okay, this analysis gives a different argument than you gave in 596, and it's a New York Times article where evidently it seems that they're conflating infectivity with severity, and cites the same animal studies that we've been debating. And so if anyone can find an example of human viral disease where inoculum size clearly affects pathogenicity, not just infectivity, we're not aware of that. But based on animal studies, as Vincent talked about last week, and I would agree with it from our own research, that when you give different doses of viruses to animals, you get a different disease from a low dose to a high dose. And it's not just a severity, it's that you get a different course of disease.
Mm-hmm. Mm-hmm.
But we just don't have human studies that show that.
I think you're right that the Times article conflates, the number of infections with the type of disease, right? So yeah, a healthcare worker is going to probably get a higher dose, and they're more likely to get infected. But the type of disease varying, we don't have any data for that. Although, as you say, in principle, based on lab studies, it should be possible, right?
And also, the Times article, if I've just looked at it briefly, is making an issue of the Dr. Lee, who was only in his mid-30s and died, and he was a physician. That's an anecdote. People in their 30s do die from this. Okay, and it's not necessarily because he got a larger dose of virus.
Rich.
Ianis writes, I was really surprised that convalescent serum got FDA approval while human isolated neutralizing monoclonal antibodies cultured in CHO cell lines against the receptor binding domain of SARS-CoV-2 are required to pass all the phases of clinical trials. I think that it is unethical to stop drugs that are like 99% likely to work with minimal side effects while dangerous procedures are allowed to go through. So I wasn't aware of this, but my guess is that there are protocols in place approved with the FDA for the use of convalescent serum.
Yeah.
And that if you produce your convalescent serum in compliance with those protocols that have been tested in the past, that you're okay to go. You're good to go. Whereas a monoclonal antibody produced by some procedure that has not been approved needs to go through the appropriate approval. That would be my guess as to what's going on. And I can't argue against that. Okay. The FDA I've dealt with the FDA a little bit. They're doing their job. It's important to test these things and make sure there aren't any problems. If you've got a new procedure for working up on an industrial scale a monoclonal antibody for use, it needs to be checked out.
So Ianis writes, these monoclonals are 99% likely to work and wants some rational thought. Well, I'll tell you, Ioannis, there has been rational thought put into it. Let me give you an example. Back in the last year, there was an outbreak of Ebola in the DRC, and 4 different kinds of monoclonals were given clinical trials in that setting. 2 out of the 4 worked, 2 out of the 4 didn't. And that's why you have to trial them. We don't know if they work. Well, you could say, well, how do you know the convalescent serum is going to work? It's from someone who recovered from infection. And it's, as Rich said, it's been used in humans many, many times. So that's the reason. But remember, the convalescent serum is emergency authorization. It's not a routine application. It still has to be subject to clinical trials. So it's not like you can just go to the pharmacy and get a prescription filled for convalescent serum.
I'm sympathetic with the idea that drugs with promise or therapies with promise need to be fast-tracked, and I'm sure that the FDA is sympathetic with that as well. But if you want to know the kind of stuff that can go wrong, think thalidomide, okay, or RSV vaccine or something like that. If you don't check these things out, not only can they work in the end, not work, but things can go wrong. So,
but you have to make sure it works, because you can't give someone a monoclonal and say this is going to cure you and then they die. That's ridiculous, right? Now, no drug is 100% effective, but you have to know that it has a good chance of working. But yeah, the fast track is in progress. I'm next from Christy. What is your outlook for the next 6 months? Cloudy with a chance of coronavirus.
Yeah.
I mean, how do you see this playing out? Best guess, well, what are we waiting for? As we isolate in our homes, I understand the point of isolation, but I'm wondering what ends and what comes after. I know we're working on vaccines, but they're 18 months away. Also, do you see this as being a flu-like virus that sticks around as one to worry about and immunize for yearly? I don't know. We don't have crystal balls, but we're thinking July-ish. This should be coming down, but we don't know. In the meantime, I think it would help for people like Christy if the leaders in our country gave some guidance instead of saying go back to work at Easter, but they're not. But I think by July, you'll probably likely be back to work, although that could change. And I think that, as Ralph Barrick has said, and as John Udell said yesterday, this is going to become another common cold coronavirus. It'll be the fifth one that we don't need to vaccinate against eventually, because kids will get infected. They won't get disease. And then when you age, you'll get infected over and over with very, very mild disease. So once this virus goes through a majority of the world's population, I don't think we're going to need a vaccine for it, but, you know.
What we don't know is how long it took the common cold coronaviruses to get to that state where they are now, because we don't know when they were like a SARS-CoV-2. One thing I would suggest is go back to the link that we put in the show last week, 595 from the Ed Yong. There were two articles, and the second one was basically a kind of how does this play out scenario that I think was well reasoned and well written. The other thing is you could take a look at the link that I put in, that we're going to put in, about the National Geographic article about how things played out in 1918 with respect to when people were released from social distancing. Did the virus stay down or did it rebound? And did social distancing have to get put back into place? And so there's some ideas that it might be that the vast majority of the country would be able to release from social distancing, but there might still be pockets. And that's why being able to test would be really good. Speaking to the reader who's saying, you know, why do we want to test? Well, we'd like to know who's got antibodies against the virus, but we'd also still like to be able to test and identify who has an active infection. I think those will all help.
I would also point out the link that I think was in the show notes last time, or I'm losing track of episodes. They're happening so fast. Maybe 96. That's this covid19.healthdata.org. That is a projection. It's a model. All models are wrong, but it's a way to think about this. And according to this model, with social distancing in place, it suggests that in the U.S. there would be a peak in mid-April, after which it might start coming down towards the end of April and being pretty much close to settled down by the beginning of June and really down to baseline by the beginning of July. we'll see how that pans out.
Dixon.
Yes. Kevin writes, I have what is likely another stupid question. I work out/exercise quite a bit, and generally when I get sick from a cold, I will tend to continue exercising, though never at a gym. I wait until I'm over it. If only everybody did the same. I like the idea that I'm keeping my heart strong and my breathing full and strong, but I've never been quite sure if that's helping or hurting me while I'm ill. I feel horrible if I just lie in bed when I'm sick. Is there any evidence that exercising while ill can be better or worse for you with any virus bacteria, not just SARS-CoV-2? Thank you. Well, and then he apologizes if this has already been covered. I try to listen to all your questions and answers, but sometimes I miss some. I can't believe that you would actually want to exercise if you're not feeling well. I just can't believe that. I certainly wouldn't. And I love fishing, but when I'm tired and achy from the day before's fishing exercise, because I'm getting a bit older, I don't go fishing that day. I wait until I feel better, and then I go back out.
Well, if he feels good enough to exercise, I think he should. I asked this of the immune crew because when you exercise, your IL-6 levels actually go up. And so the question is, it's that bad? But they said there are more than IL-6 involvements in the immunopathology of SARS-CoV-2. So their consensus was, if you feel well enough to exercise, I think it's okay. However, I am not a doctor.
Right. I think for anything that you do exercise-wise at the gym, if your body needs you to stop, it's going to tell you that. So that's the kind of byword that I would use.
Yeah, I think just listening to your body is the important thing here. It sounds like you're pretty much in touch.
Dixon, you listen to your body?
Absolutely, boy, and it's telling me all kinds of things nowadays. But I used to run every other day, and I used to run like 5 miles. When I didn't feel good, I didn't run.
You ran a marathon, didn't you? Didn't you run a marathon, Dixon?
Yes, I did.
And how many hours?
4 hours and 23 minutes.
It's a good job. Very good. Kathy?
And without stopping.
Robin writes, I live in an apartment building with probably about 100 to 200 tenants and was curious about transmission within apartment buildings. Any guesses about how much transmission risk there is from apartment to apartment and how much it can be reduced? I'm also wondering how you'd estimate the contribution of hospital overloading to mortality. I've heard a lot of noise about plans for ventilator rationing, but not much about whether it's actually gone into practice. And I've heard of ICU wait times in some hotspots, but I don't know how much that actually affects outcomes. So we're not sure about risk of spread within apartment buildings. There seems like there probably is some risk, but we haven't seen any data about this. And one suggestion might be to minimize your time in public spaces of the building and wash your hands when you return to the apartment. For instance, if you've been in the elevator or to the laundry or to the trash chute or dump or whatever. Hopefully some expert in hospital management could chime in. We have had some input on architecture of hospitals, that there is a whole science, if you will, about that. But with respect to the specific hospital overloading, I have seen articles too about the possibility of having to ration ventilators and so forth. And I go back to the top of the show when Daniel talked about that, and what they're hoping to be able to do is put 2, 3, or 4 people on one ventilator if it gets to that point.
I think this is the whole flattening the curve thing. The whole reason we're trying to flatten the curve is to prevent the overloading of the hospital, because if that gets too extreme, it is going to impact mortality to this disease or others that people need hospitals for.
So I might be the only person that lives in a similar building of the 5 of us that ordinarily podcast here. So our building management has been very good at giving us heads up and reminding us that we shouldn't dwell in the lobbies and talk with the management people, like the doormen and people like that. Wash your hands every time you go out to take out the trash or to do the laundry. Just assume that the virus is everywhere and just do the right thing. And so far, so good. So I think we haven't had any positive people in our building. They promised to let us know if that happens so that we could take extra precautions. We have a lot of older people that live in this building over the age of 70, upwards of 95. So we have a very susceptible population. And so far, so good.
Does anyone know, speaking of rationing vents, so Daniel was complaining that these hospital ships were empty. Does anyone know why they're empty?
I don't understand that, because I thought that I meant to ask him about that, because I thought the idea was that the hospital ships were going to be populated with people who did not have COVID. Keep the COVID off the ships. And Daniel indicates that there had been some problem.
Well, the problem is that now, if we're going to assume that everybody is COVID positive until proven negative, then you still have a problem with putting people on a ship that you think are there just because they broke their leg. They might be COVID positive as well.
Think of how many people it would take to move a single person out of a hospital, transport them, get them onto the ship. There's a lot of personnel there, and I think those people are already spoken for right now.
All right, let's do one more. Rich.
Mike writes, sending this relative to Rich's comment about in TWiV 596 about trying to determine how this ends, and Alan's subsequent comments about how the world is now watching China. The linked video is from a British visitor to Shanghai. It shows that as of March 18th, the lockdown isn't so much lifted as partially relaxed 2 months after it was implemented. People are out and about, but in smaller numbers. Restaurants are still closed for sit-down dining. Internal movement around the country is still heavily constrained by a cell phone app that gives each person a status of green, yellow, or red. Red means you are restricted from driving in many parts of the country. China's current approach relies very heavily on their pervasive domestic surveillance apparatus. The maker of the video has some very guarded comments at the end about the darker sides of this system to avoid placing his Chinese friends in jeopardy. I thought it was revealing and wanted to pass along. Personally, I hope democratic nations can implement something closer to the South Korean model. So what I take away from that, among other things, is that, we said we're now watching China because they're lifting restrictions, but it sounds like it's not as if all of a sudden everybody can move around and travel anywhere. So, we should watch them, but we should also understand the extent to which they have actually lifted restrictions in order to understand the impact of that.
Okay, that'll do it for TWiV 598. You can find it on any podcast player. Please subscribe so you get every episode. It's free, and you know when we release a new one, which has been pretty frequent lately. And we hope you enjoy the programming. You know, we're just trying to get interesting people and ourselves to answer your questions. If you want to look at the show notes, which has all the letters, all the text of the letters and links, microbe.tv/TWiV, questions and comments, TWiV at microbe.tv. A number of people have said, I can't find a link to send you an email. Well, it's at the end of each episode. Sorry, you have to listen. It's also on the website. If you like what we do, consider supporting us financially. Microbe.tv/contribute. Our guests today have been, of course, Daniel Griffin, out in the trenches taking care of patients. We thank him very much for being here. Dixon Despommier at trichinella.org and parasiteswithoutborders.com. Thank you, Dixon.
Pleasure.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is good.
Rich Conditz, an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time. You know, we got through all of 15 pages out of 129 of email. If we hung in there for another 10 hours or so, we could get this done.
We're doing our best, yeah.
Yeah. Well, it's good. It's all good.
I'm glad that, you know, we do a couple a week and some people join me, and I really appreciate whoever is able to do it. And I I know it's a lot of stuff, but we're trying to, where else can you go to get this stuff discussed? I don't think there are many other places. So we're happy to do that. We love it. I'm Vincent Racaniello. You can find me at virology.blog. I want to thank ASV. That's the American Society for Virology, by the way, and ASM for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.